Hi everybody, welcome back to GenUX. Today we're talking to David, who is not just a professor, but he's also one of my friends. He's actually currently my neighbor. And we got to talking a couple weeks ago about a lot of things that he studies, which is fertility within cows and how it's transferred from not the cow embryo, but how the information is also transferred in parallel to human beings. So without further ado, David, tell us about you.
Yeah.
And where you're from, how you got into this business, and where you work, and all the good things.
Yeah. My name is Dawit Tesfaye. I'm born and raised in Ethiopia, so in a small town called Jijjiga, where I visited schools. And later on, after completing high school, I joined one of the agricultural universities in the country, that is Aramaya University, where I studied my bachelor in animal science. And after doing that, after graduation, I joined the Ministry of Industry, working on several projects. And then later on, I was lucky to have a scholarship to proceed with my master's in Germany. So I joined the University of Bonn in Germany, and I studied my master's, and then I did my PhD there. And then later on, I was a faculty member at that specific university. So that is the journey before I came to US. And then later on, in 2017, I had a chance to do a research scholarship, or research scholar or sabbatical at the UC Davis, where I started to work on gene editing in pre-implantation embryos in Pablo Ross lab. And then later on, the position here at CSU was announced, and then I competed for that. In October 2019, I joined the Colorado State University as associate professor. I'm still working on that. That is where I am now.
So what is your current title, and what is your lab called here at this college?
So I am in the College of Veterinary Medicine, Biomedical Sciences, in the Department of Biomedical Sciences. So my lab is located here at the Foothills campus in Animal Reproduction and Biotechnology Lab. This is a place for some of the faculty members working on in animal reproduction. And my lab is also located here in Animal Reproduction Biotechnology Lab. So welcome everyone. So this is just a picture showing that what our lab is focusing on. So that is the so-called the pre-implantation embryo development period, which we are interested in to analyze and to see how embryo development from the oocyte stage to the blastocyst stage is regulated. And we see here that when we get ovaries from the slaughterhouse, mostly we get our ovaries from the slaughterhouse, when you aspirate them, and you find a lot of cell dermis and also some oocytes with surrounded by cumulus cells. Once we select the best ones, and then you will have the immature oocytes, which are not yet matured. When they mature in vitro, then you will see the expansion of the cumulus oocyte complex. So the cumulus cells will be expanded. After fertilization, the first embryo will be formed. That is called the zygote. And then the first cleavage is happening, and then we have 2 cell, 4 cell, 8 cell, 16 cell, and early blastocyst or morula, and we have the blastocyst and well expanded blastocyst. Then also the one which is starting to hatch here at this point is going to hatch, and then we see that blastocyst. So this is all what we are using as a model to understand what determines or what molecular mechanisms are responsible for getting from here to here, and what determines the ability of this blastocyst to induce pregnancy after transfer to recipients. So this is all over about that. So we are producing embryos here. We are doing several experiments. These experiments are all designed to understand molecular mechanisms associated with embryo development and embryo developmental competence.
So for the people at home who don't know much about this fertilization process, can you tell me from zygote to blastocyst how many days that normally takes in the cycle?
Yeah, this is the first day after fertilization. Then this is the day 7 to day 7 expanded blastocyst stage. That means a 1 week period from that stage to this stage. Mostly this development is happening without any increase in size of the embryos. They just divide, multiply, divide, multiply, and then after a certain time, they differentiate into two cell populations. This one is this one is, for example, the inner cell mass, which becomes later on an embryo, and the other one, which the cells which are surrounding here, are the trophectoderm cells. They are making the placenta. So this is the first stage in which cell differentiation happens into two cell lineage, and that will go on and develop into fetus, embryo, and the placenta. So this is a 7-day developmental window which we are interested in, because of the fact that, why are we interested in these ones? This is because of the fact that both in human and also in livestock, fertility is mostly associated with because of high embryo mortality in the first 6 weeks of development. So to tackle fertility, then you have to understand what derives why some embryos are surviving while the others are dying under certain specific environmental conditions.
So can you also explain the difference between, it looks like day 3, when you have 8+ cell division to blastocysts, how important that moment is in the fertility process?
Yeah. So the various events which are happening during the pre-implantation period are dependent on, first of all, development until the 8 cell stage is dependent on the maternal factors which are stored in the oocytes during the maturation process. The RNAs, the proteins which are stored in here are supporting development until here before the embryo is genetically active. That means there will be a shift from the maternal control of development to embryonic control of development. This shift is very critical, where most of the embryos are dying at that stage because of their difficulty for them to shift from the maternal control of development to embryonic development. Once the 8 cell stage is passed, then embryo is dependent on its own genetic activity. That's why development after this is much more easier and differentiate into different types of cells, as you can see here.
And this is true for humans and
animals. Exactly. Okay. Especially the bovine and the human are having a similar transition from maternal to embryonic genome, because in human, in bovine, this transition from the maternal to embryonic genome activation is happening at 8 cell stage. In mouse, it's earlier in 2 cell stage, but in human and bovine, it's more or less similar. 8 to 16 cell stage is the one which shifts from the maternal control to embryonic control.
Very interesting.
Yeah. As I said, we are generating our embryos in vitro as a model to understand, to model the embryo development in vitro environment. So we get ovaries from the slaughterhouse of cattle ovaries, and these ovaries will come every 2 times a week, and students are getting them. They aspirate the oocytes, and then they aspirate the ovaries, and they collect the oocytes, and we let them mature for 24 hours in maturation media in an incubator like this one.
Okay.
So, and then afterwards, what will happen is that after fertilization, after maturation, the next day, the oocytes will be fertilized. So we use a bull sperm to fertilize them under in vitro and the petri dish, and that after fertilization, 24 hours later, those embryos, those cells which are around the oocytes will be removed, and the zygotes will be further cultured in the culture media. And those culture medias are 2-step based on the nutrient requirement of the embryos up to a certain stage, up to 8 cell or morula stage. Embryos are dependent on specific glucose or whatsoever nutrients. So that media is designed in such a way that it will meet the requirements in a stage-specific manner. So once we get that one, so the embryos are produced for specific experimental setups. For example, if we are using antioxidant supplementation, then that specific oocytes which were derived from those ovaries will be used for that specific experiment. And some of the other experiments are working on heat stress model to see the impact of heat stress on embryos. So we have also another work which we can discuss later on, which we are working in collaboration with the Colorado Center for Reproductive Medicine, where one of my PhD students stationed there to work on maternal age on impact on ovarian function in oocyte development. So these are the things which we are doing here.
That's awesome. So tell us about some of the machines that are behind you.
Yeah. I mean, this is a monogas incubator, which we are using for mostly for oocyte maturation, granular cell culture, things like that. This is a clean workbench where we are working on media preparation, manipulating of the cells and everything, what we do here. And this is a tri-gas incubator. Once fertilization takes place, all development of embryos is happening in this tri-gas incubator, which is adjusted with the oxygen tension, which is similar to the mother. That means the in vivo. That means the 5% oxygen tension is what is available in the reproductive tract. So to simulate that, this tri-gas incubator will be used, and embryos will be checked every time for their development, for the stage of development, for anything under microscope what we have here. So depending on the experiment what we are doing, everything is arranged in such a way that that specific culture environment is answering a question which is asked by that specific experiment.
We're at CSU's lab today, just to let everybody know where we are in the world in Fort Collins, Colorado. So if anybody's interested in finding out more information about DeWitt, this is one of the places you can go to find it. So how did you get into this field? Like, how did you start from, being from Ethiopia, getting your first scholarship, and what about this specific thing really set you on fire and made you interested to do what you're doing?
I mean, originally when I started to study animal science at the bachelor level, so that was just an interest which is ignited by, Ethiopia has the largest cattle population in Africa, I think 10th in the world. So that forced me or interested me just to deal about animal science. And then later on so initially my interest was animal production. But after I joined the University of Bonn, after I do my PhD, during my PhD time, I joined the lab which was working on reproduction. So that, starting from that time on, my inspiration for reproduction and reproductive biotechnology is there. And then I've continued with that one until now. So my research, which is I'm doing here at Colorado State University, still is a continuation of the work which I did at the University of Bonn in Germany.
That's really cool.
Yeah.
So a lot of people don't know how a lot of these labs get funded or how professors continue to have money to do this type of research. So can you talk to us a little bit about how the lab itself gets funded, how you get funding, and what you have to do every year to maintain that funding?
Yeah. I mean, it depends on the line of research what we are doing. We have two interconnected direction of research. One is researching to improve the reproductive efficiency of the dairy and beef industry through reproductive or assisted reproductive biotechnology procedures. The other line of research is to generate knowledge from those research which can be translated to human reproductive medicine. So when we target research on improving the reproductive efficiency of animals, then we target the United States Department of Agriculture. The USDA is the main funder. And the other side is the translation of knowledge from the animal model, what we are using, to the human reproductive medicine. Then we are targeting the National Institute of Health, or NIH, as a funding source for our research.
That's really cool. So how is the data that you've received in your lab, and you've mentioned that it translates to human beings, like how is that correlation seen through data? How have you best explained it to the people that you get funding from, and to your students and to your PhD members in your lab? How does that all work together?
Yeah. The pre-implantation period in which we are interested in how embryos are developing, how embryos are responding to the maternal or any type of environment, why embryos are dying, why some embryos are dying, why the others are surviving and inducing pregnancy. This phenomena is more or less conserved between animals and human, especially when it comes to the, the cattle as a model for human, then it is more related to human than a mouse model, for example, in various aspects of reproduction, in terms of their giving birth for a single offspring, like human, in terms of their length of gestation, it's almost 9 months the same way. In terms of their follicular dynamics and everything, cattle are more closer to human than other species. Yeah. But the other species advantage is, for example, the mouse advantage is you have a chance to understand some mechanistic insights in detail, because you have a chance to generate some knockout models to understand some mechanisms in very detailed way. So that all is the core for me. And the other thing is that in the current times, so assisted reproductive technologies, which are targeted to improve fertility problems in human and in animals, they are all in both directions. It's increasing. In human, the fertility clinics are using a lot of IVF procedures to help couples to conceive, especially when they come to the clinic because of their failure to get pregnant naturally.
Yeah.
So they get help through assisted reproductive technologies, mainly the in vitro fertilization of embryos. In the same way, in the dairy and beef industry, the dairy and beef industry is highly relying at this moment on transfer of embryos produced in vitro from superior cows to advance, to shorten the generation interval and advance genetic gain in the field to improve fertility in the dairy cows. So assisted reproductive technology is targeting fertility problems both in human and animals, so that we will have a common target for both of them.
So in the studies that you've been doing, have you been, first of all, that's really interesting. I didn't know that the industry was also very interested in having the strongest genetic creatures reproduce for the other strongest genetic creatures, which I mean, if you think about over time, when humans were evolving, and the ones that had the babies and were able to push babies out, not the ones that died in childbirth, we are now the stronger species. So I mean, it kind of rings true both ways. So we also talked about when we were in your lab, we were talking about data and how you get grant money and all the information that you're collecting. And we kind of, at one point we were talking about, when you are doing research for specific grants, you have a hypothesis for that grant, and that's how you get funded. But you do find other data during that research. And so sometimes what comes out of that data that you find that's, I would call, in my world, it's called opportunity data. I don't know if it's called that in your lab, but it's things that you weren't necessarily asking, but trends that start to develop as you're getting data back, and you notice all these new pieces of data. What do you guys do with that?
Research is sometimes is a kind of exploration as well, through the whole processes what we are doing. But as you say, that results are there for us just to generate data, to see the results. Some of the results are fitting to the theories what we have, or the hypothesis what we have. Some of the data may not be fitting to the theory. So for those which are not fitting to the theory, what we do is that we just see first why is that, and we need to see the reproducibility of those results. We need to repeat them controlled way, and then see that finding is repeatable. So if that is reproducible, then that is a good thing to see into the result. And you can, with those results, you can reshape your hypothesis, you can refine, and that will lead you closer to the truth of the main problem. So reproductive biology in specifically, and also biology in general, is not a static thing. It's a very dynamic. It's most of the things what we are investigating are very multifactorial. So fertility or embryo survival, embryo death. These are all um, uh, multifactorial. That means they will be affected by several factors. Because of that all, there will be sometimes some surprises, but surprises are always there. Surprises are good as long as they are reproducible. They are a good source of information which can be used to reshape our hypothesis and also to think in a different way than we thought before. That means we go a different direction. Otherwise, if the hypothesis is supported by a result, what we get, that it will be a preliminary data, what we are using to draw another hypothesis, which we go with that hypothesis and grant to the funding agencies.
Right.
So that means the funding agency wants to see what kind of preliminary data, what we generated, and how we design or generate our hypothesis from those preliminary data, and what we are going to test in that experiment.
Another thing that we kind of talk about in research is, when you talk about hypotheses and you talk about theories and you talk about how when you have a hypothesis or theory and you're testing to it, the results have to be duplicable. They can't be like standalone, N of one, this thing really cool happened and wow, like this thing happened. Let's focus on that. That's not what research is. What research is stating a hypothesis, explaining how you're going to support the hypothesis with data, and collecting that data, and for those people who don't know, reporting that information out. So in a multifaceted situation that's dynamic, which is fertility, because there's a lot of different things that can affect fertility, what challenges do you find when you're going through your process here at the lab, or just an everyday kind of thing that you notice that you do that's challenging?
Yeah, I mean, one of the challenges which I can say is the bridging the gap between the data what we generate in vitro and the things which are happening in vivo. They are two different things, and that bridging that gap is more of a challenge. So, for example, the central point for our research is the embryos intrinsic quality is the one which determines the embryo's ability to induce pregnancy after transfer. And then the question will be, the challenge will be now, what are the different factors which compromise those capacities of the embryo? The maternal age, the lifestyle, nutrition, environment, heat stress. These are all factors which are contributing to the embryo's ability to induce pregnancy. That means the quality of the embryo is what we get out of that. So those gaps, bridging the gap between in vitro and vivo, that's one thing what we are trying to do, because of the fact that, for example, in vitro produced embryos are developmentally compromised sometimes compared to the in vivo derived ones. So there are some factors which are missing in the in vitro environment compared to the in vivo, compared to the in vivo. For example, when you do IVF, what is the central point? Which is missing is this the tube-like structure between the ovary and the uterus, where the pre-implantation embryo development is happening. That's called the fallopian tube in human. That is the oviduct in the animals. So this is the window in which we are doing the in vitro fertilization in the lab without those oviductal factors. So what we are doing at this moment is just to we hypothesize that all these negative consequences on the embryo produced in vitro is because of the missing factors which are present in the oviduct of the embryo, where fertilization happens, where the first cleavage happens before the embryo goes into the uterus. So what we do is that we just try to develop a so-called organoid technology. Organoid technology is a kind of mimicking the organ using a 3D culture system, using the epithelial cells from the oviduct. That organoid are structurally and functionally mimicking more of the oviduct, and we manipulate them, or manipulate means we treat them in such a way, we treat them the necessary hormones and things like that, so that they can release factors into the culture media in vitro, which can compensate for the missing oviductal factors in the IVF system. So that is one of the research what we are doing. And that is the target for us to bridge the gap between in vitro and in vivo.
That sounds hard.
Yeah.
That just sounds
Yeah,
hard.
yeah, I know, but it is.
But that's a really interesting field of study, though. So that would be like, that is challenging. I mean, to figure out how to do that. I mean, I'm familiar with IVF because like I've told you, we've gone through the process, and we personally never made it to blastocyst, which is why we talked kind of a little bit about blastocyst openly and honestly. Is there anything that you do right now that just really excites you on a day-to-day basis? Like anything that you're working on at this moment that just like really wakes you up in the morning and makes you excited to go to the lab?
Yeah, I mean, one of the opportunities in our field of research is, the continuously advanced level of analysis what we are able to do. Nowadays, you can do single cell RNA-seq, you can do single cell attack-seq. Just molecular analysis at single cell level will allow you to, to have the knowledge, not from the global perspective, but also at a very detailed level, which you have a chance to see molecular, mechanisms associated to a certain process at single cell resolution. That is one of the exciting things what we have. And the other thing is that this development of organoid technology, just to have opportunities for, IVF procedure to generate factors which are missing in the IVF procedure to compensate for those missing factors in vitro. The other one is this, the other area of research what we are interested in is the extracellular vesicles. These are vesicles released from cells in our body, fluids, any fluids, urine, sweat, saliva, milk, whatsoever. It is full of these extracellular vesicles. These are vesicles which are containing various bioactive molecules, RNAs, proteins, DNA, whatsoever, which are important for communication between cells. Especially in our case, the communication between the embryo and the mother is very critical, starting from the ovaries, where we have the ovary having the follicle. In the follicle, in the fluid, is an oocyte, which is surrounded by various types of cells. And that communication early on is established through these extracellular vesicles. So the detailed analysis on extracellular vesicles, the technology or the organoid technology which we have at hand, and the ability to do molecular analysis at single cell resolution is making science very attractive and exciting and very, very interesting.
I think it's interesting. I mean, I could talk to you about this all day, but we have. So are you teaching currently? Like, I know sometimes teachers just, their grant covers all of that. So do you actually
I'm teaching currently, I'm teaching human and animal reproduction at the BMS 409 level. So this is taught by two instructors. The first part is covered by a colleague at the moment, and I will be starting from beginning of October to the end of December my time, and there I will do the teaching for graduate and graduate students in the field of animal reproduction.
And so let's say some of those students were really interested in what you're teaching and they wanted to work with you in your lab. Like, what's the process for them to be involved in your lab?
I mean, one of our lab mission is, generating the next generation or training the next generation scientists. So for that, I'm the one who is talking now, but most of the work is done by the students. So graduate students, PhD students, and postdocs. So we are always open for that as long as there is a resource for that. That means we need to have grants and everything, which allows us to hire students so that they can come. Some of the students are coming with their own funding, but we provide them with all the necessary logistics and financial support to do the research. So they come, and there are different projects in the lab, so they will be involved in one or the other projects which are running. So they will ask specific questions which belongs to the overarching hypothesis.
Mhm.
So they take part of that and they work on that one. They can do their master's degree. They can write their master thesis on that. They can do their PhD theses. Postdocs are producing their um, their um,
Their
their
papers.
papers and whatsoever. They are preparing themselves to be independent faculty members in the future. So in my career, I have trained almost 12 PhD students, 6 master students, almost 3 to 4 postdocs during
Oh, that's
my whole
awesome.
time at the University of Bonn and here.
That's a lot, by the way, for people who don't know that. That's quite a few success stories coming out of one person's lab, or if
Yeah.
one person's. I mean, I know you've been in 2 different labs, but
Yeah.
coming underneath you, that's a lot.
Yeah.
Can you say somebody sees this and they're like, "Wow, this is really interesting research. We want to help fund that." Is there a place for them to go and fund it?
Yeah, so as I said, that our funding sources are mostly the federal funding from the NIH or from the USDA, where we constantly submitting grants to get funding. We were successful in the last years, and also we are also submitting some of the training grants to train students. So with that, this is a National Needs Fellowship program, which we have uh, which we got a grant from the USDA just to train 3 PhD students for 5 years and generate the next, so teach or train the next generation animal scientists for leadership. So that's what we have done, is still going on. Otherwise, some of the industry partners are also interested in collaborating with us. Mostly, when I was in Germany, I was collaborating with various industries like the Pfizer or Evonik, who are interested on some aspects of fertility in livestock. So that is the way how we are also funding our research from
So
private
public and
industries.
private, yeah.
Exactly.
So can just individuals, say somebody who, like a donor for the school or someone who really liked this specific lab, could they just donate money to you too?
Exactly,
Yeah?
exactly. So
Okay.
the university has an office which is responsible for that purpose too. And also, if they are interested in my work, they can visit my website and also whatsoever what I have in social media, so they can see if they are interested in that one. They can directly contact me or the university just to make sure that they have the funding for that specific research.
So if you're interested in learning more, you can go to Dawit's website, which is?
Yeah, that is when you go to the Biomedical Science Department website, there is a faculty list. Under the faculty list, there is my lab website, where you can have a chance to see all the the research what we are doing, the activities what we are doing, what our mission is, what our goal is in the whole fertility research.
That's awesome. And you'll see it below in the links. And all you have to do is go and check out Dawit in the links below. Thank you guys so much for joining. We were so happy to have you with this series of intrinsic data, but this is a little more, this is less intrinsic. This is more like in your face kind of data, but it's a really interesting lab just to consider.